Hello, everybody. Welcome to Ophthalmology Mastery Clinical Skills for MBBS students. In this video, we'll learn about color vision tests. What are the different color vision tests and what you should be knowing the basic information about color vision tests. Color vision is basically an ability of the eye to discriminate between different colors excited by light of different wavelengths. Okay, so it's the ability of the eye to discriminate between different colors. It's basically a function of cones. Rods do not have the ability to discriminate colors. So it is so the cones function in bright lights, right? So it is appreciated in bright light or photopic vision. In dim light or photopic vision, all colors are seen as gray because it's not a function of rods. This is called as Purkinje image. So a red flower will appear in hues of gray during in dim light, and this is called as Purkinje shift. Right, so what are the color vision tests? So we have around 7 color vision tests: Ishihara's chart, Edridge green lantern test, Holmgren's wool test, Farnsworth Munsell 100 U test, Farnsworth D15 hue discrimination test, City University chart, and Nagel's anomaliscope. Of this, Ishihara's chart is the most important, and you need to know how to do it and how to interpret it, because this is one of the competencies that you have to know, right? So the color vision chart first is the Ishihara's chart. Ishihara's test, we use a Ishihara's chart which has almost 24 or 36 plates. The patient is seated at the chart is held at 30 centimeter. The reading glasses are put on and it should be in a well lit room. Okay, so you will examine one eye at a time and you will ask the patient to read each chart and you will record it, what the patient sees and what is the chart. So a normal person sees what is present. So basically Ishihara's chart is random dots, okay, against of primary colors against dots of different color. So basically it differentiates, I mean identifies red green color deficiency. So a normal person will read some numbers, whereas a person with red green deficiency reads something and a person with total color blindness will not read anything. Okay, so you can identify a proton or a deuteron. So you will only identify red green color deficiency in case of Ishihara's chart. Okay, you cannot identify blue yellow deficiencies. Now Edridge green lantern test. So in this test the two lights are presented in a vertical display and it can be in any combination. So the patient has to say first is above red, above green, and below red, above white or green. So this is the combination, and if the patient passes 8 out of 9 times, it's called the pass. It is said it is pass. Okay, so it is basically performance based. It does not diagnose, classify, or grade the level of color vision defect. It only says whether patient has color deficiency or not. Okay. Then the next one is Holmgren's wool test. So here, the patient is asked to match skeins. There's a selection of skeins of colored wools. So he has to match green with green. So you give him something and he has to pick up another skein from the box and say this is matching. So that is, so again here you can see that only gross color deficiency can be identified. Does not classify or grade the level of color vision defect. This is also for usually red green color deficiency. Farnsworth Munsell's 100U test. This is the gold standard at the highest sensitivity and specificity and can identify all the color deficiencies. So basically here, though we call it as 100U test, it has only 85 chips and arranged in 4 rows. Okay, so the patient is asked to arrange the colored chips in ascending order of hues. So if he starts from, and he has to arrange the chips in the ascending order, and this permits both classification and severity of the type of color vision deficiency. So basically it's an error score. So he's asked to do it and then you see how many mistakes he has made. So the higher the score, poorer the color vision. So lower the score, it's a better color vision. So it's the most sensitive and can identify congenital deficiencies, acquired color deficiencies, can identify red, green, blue, yellow, everything. It can identify everything. But the disadvantage is it's time consuming. It takes a long time for each patient. So next came up the thing of Fansworth D1500 UTIS instead of 20, four chips, four lines. You have only one line, which is 15 hues. So patient has to arrange them in 15 in the ascending order and estimates all the color deficiencies again, but does not estimate the degree of deficiency. It can only tell you whether it is there or no. Okay, and it is faster than the 100 U test. The next is a city university chart. It was started in New York. So here you have four colors from are surrounding the center colored plate. So you have a central colored plate and four colors around it. So patient is asked to match the closest hue from the center one. He has to compare with the four hues in the surrounding thing and say which one it is closest. This is only 10 plates. Again, does not indicate any degree of deficiency, but it is faster. So usually it's used in some industrial before joining any industry, whether they to crossly identify any presence or absence of red green color deficiency. The last one is Nagel's anomaloscope. Here the patient is sees through the anomaloscope. He sees one half is yellow, completely yellow, and the other half he can mix and match the red green. So two knobs are given, and he has to match them until he sees that both are yellow. Okay, so he is allowed to match them on, and here the observer must mix that. So both knobs, one mixes the red and the one gives the green. So he has to adjust the knob by himself until he says that it is matched. Based on how much the thing we the scoring is done, and it can match the proton knob or the deuteron knob. Okay, again red green color deficiency can be identified in this. So what are the congenital, those are the tests. You also need to know a little bit about color deficiencies color defects. They can be dyschromatopsia or achromatopsia. Dyschromatopsia is relative absence and achromatopsia is total absence. In dyschromatopsia, you can have anomalous both of them. You can have anomalous trichromatic color vision, dichromatic. So anomalous, they have all three, but then one of them is defective. So protanomalous defective red, deuteranomalous defective green, tritonomalous defective blue. Dichromatic color vision, one is totally absent. So protanopia absent red, only they can appreciate only green and blue. Deuteranopia absent green, tritonopia absent blue, or both red and green are totally absent. Then they have only blue cone monochromatism. It's called as blue cone monochromatism. Here it's usually associated also with poor day vision and visual acuity is reduced. So in these anomalous trichromatic color vision and dichromatic color vision, the visual acuity might be still be good, 6/6 vision, but they have just the color vision is defective. Whereas in blue cone monochromatism, color vision is also reduced. Right, so in achromatopsia it's usually total rod monochromatism. There is total color blindness, so cones are not really functioning. So when we say cones are not functioning very well, there is also day blindness. So there is visual acuity is usually reduced. The patient might have nystagmus, but the fundus might be normal. You will not be able to appreciate any pathology. Acquired color deficiencies, there can be only two types: blue yellow impairment or red green impairment, and usually they are unilateral, and the visual acuity is also reduced. The visual acuity reduction is proportionate to the color vision loss in these cases. Unlike in congenital deficiencies where the visual acuity might be good and only color vision is reduced, in acquired color deficiencies the visual acuity is also reduced. Blue yellow impairment, please remember macular lesions. Red green impairment, optic nerve lesions. So any macular lesions like central serous retinopathy, macular edema, they might say that the blue yellow spectrum is defective. And especially if it is unilateral, they can appreciate better, and they say in one eye I see a different color, in the other eye I see a different color. Red-green deficiency in optic nerve lesions, again optic neuritis, Leber's optic atrophy or compression of the optic nerve, you can see red-green color impairment. So in color vision test, you might be asked how to perform a Ishihara test. You might be asked to enumerate the different types of color vision tests and a bit about color vision congenital color deficiencies and acquired color deficiencies. Thank you very much.